I'm going to talk today about some of the work my group has done in sort of machine learning for early stage drug discovery. And I put some of our papers up here, and of course you'll be able to look at the talk later so you can look through them. But it's really to give you a flavour of the kinds of approaches we've been looking at. It runs through from predicting whether molecules will bind or not bind to a given protein target. So if you look at the bottom right there, to trying to remove biases from that kind of work. And then finally, how do we generate novel molecules, but in the protein binding site? So can I generate chemistry that is useful for a particular protein that I am interested in using these kind of machine learning techniques? So the first piece of work I'm going to talk about is for virtual screening. It's a method that we call DenseFS. Virtual screening is a really well-established problem in drug discovery. So what I have is the structure of a protein. So here, that's kind of the surface you can see, so the red and blue stuff. And that might be a crystal structure, it might be an NMR structure, it might be a homology model. And then what I'm going to do is stick lots of small molecules in it, so ligands, like that white one that I've got in there, and I want an answer to a very simple question: will this molecule bind or will it not bind? And it sounds like an easy question, because this is a question of kind of the physical interactions between these two objects. We kind of understand the chemistry and the physics here and what should make things bind and not bind. Turns out we're actually not that good at it. Standard techniques until very recently was to perform docking. So in that, I would try and dock this small molecule into the protein, and I would have to give some amount of flexibility if I possibly could, and say, well, do the kind of chemical interactions that are made look good? So do I think this will bind or will not bind? Recent has been the change to move towards thinking of, can we use statistical functions to learn this? And there were just sort of standard statistical functions to learn this, uses of things as simple as things like linear regression, and then moving on to much more complicated machine learning methods. And the most successful of these so far has been using these convolutional neural nets. And I'm going to talk through how we've used them and why I think they've been successful. Now this next slide looks like I've jumped completely sideways out of proteins, and I'm not interested in doing drug discovery anymore at all. But it's to try and explain why people went to the convolutional neural nets. Now the area that machine learning really kind of took off in, and everyone started to see that it could do things that people didn't think were possible with these techniques before, was in image recognition. So there was a competition launched in 2010, and it actually stopped happening in 2017, because by 2015, the computers were better at recognizing images than the humans. So the task here was quite simple. It was a set of pictures, and the computer had to say, does this picture contain a cat or a dog or a bird or a plane or a truck? And these kinds of CNNs, and the deep CNNs really started in sort of 2012, overtook the ability of humans to do this. So the idea here is I want to do that same kind of image recognition, use what I know these tools are powerful for, but use it to answer a different question, which is bind or not bind. In order to do that, what I'm going to do is turn a protein binding site into an image. So here we're going to think of it as a 3D image rather than a 2D image. And in an image, your channels in each pixel of an image are your red, green, and blue. So you can imagine in each pixel it says this has a certain percentage of red, green, and blue, or it might have 0 of all of them. So instead of that, I'm going to have a set of, in this case it is 34 channels, which are atom types. So for example, how much aliphatic carbon is there? And now it will be in a voxel, so it will be in a 3-dimensional box, a voxel. In that box, how much nitrogen acceptor, how much sulfur, and so on through this. And so I end up with this kind of image of the binding site. And now all I want to do, and it's a very easy question, is I'm going to say, well, does it bind or does it not bind? And that's what I want the thing to learn. So I will train it with ligands that bind and with ligands that don't bind. And then when I give it a new ligand, hopefully it will have learned that. The important things I want to sort of remind you of here, which will come back through the talk, is I'm not going to tell the model anything about interactions. So I'm quite literally going to tell it atom types, describing where they are, and that's it. So I'm not telling it about how they're interconnected in the protein. I'm not telling it about the chemistry of the ligand. All I'm going to say is there are these ligand atoms near these receptor atoms, and that's all the information it will have. I'm going to feed that into a deep learning CNN. The one that we used is the one on the bottom here, which is kind of at the level of some of the more recent winners of the ImageNet competition. I put the one on the top there because we weren't the first people to do this kind of work. So David Coze's group did this published it in 2017 with a much simpler convolutional neural net. It turns out that making, if you like, the improvement that has been seen in the architectures in terms of image recognition also carries through, because you're trying to use them for a very similar thing when we use them for this kind of prediction of bind, not bind. But there are other steps we made as well which changed the way that this worked. To do this, I'm going to start with what is a very sort of standard set. So the DUD-E set, the database of useful decoys. I'm going to use this for training and cross-validation. There's 102 protein targets in that, and there's over 22,000 active compounds known and 1.5 million decoy molecules. When I'm doing my cross-validation, my train and test are always separate, so there is no target that would be in both my train and test sets. So one of the issues with this set, and it's important for everything that happens from here, is that the true poses are unknown. So what we have to do here is we have docked everything. So I have taken all of the active molecules and all the decoy molecules and docked them in. I am always going to train using the top ranked docked pose, which may or may not be correct, and I will talk a little bit about how quite often it isn't correct, but I might test using that top ranked docked pose or all the generated poses. So previously people had always just taken the top ranked one, but there's no reason why I can't look at all the poses, because once these models are trained, they're incredibly rapid. So why would I want to test on more than just the top pose? Well, this is a very nice example of this. So this is the top docked poses of a particular DUD-E target. And the Wiener dock pose is in the top left of this picture, but the actual crystal pose would be in the center. So this is something where we do actually know the structure. And in this case, we know that it should have docked in the middle and it didn't. And if you like this kind of conglomerate of like blue bits you can see all over the purple structure, these are all the different docks that were suggested by the Wiener docking software. So does it work? Well, this is just cross-validation on the DUD-E set. This is a precision and recall curve. So if I was perfect, which obviously I wish I was, but in this case I am not, I would want to be a horizontal line that ran across from that one in terms of precision all the way across the top of the graph. So I had 100% precision and 100% recall top right-hand corner, and I'd like it to be a straight line across there. And the other way of measuring this would be the area I got under my graph, which would obviously then be 1 because it would be the whole box. If you look at the red dashed line at the bottom, that's random for this task with this data set. So everything I'm showing you on here does significantly better than random. The blue line is how well standard docking software does at the task we've just set it. So if I give it the decoys and I give it the actives, how often can it actually tell that I've got an active? Well, it can, but it's not great. So even when I have very low recalls of only about 10%, I'm only getting it right about 20% of the time. The black line, which I call baseline CNN, that comes from David Coze's work. So that's the earlier versions of being able to do this. The orange line, that is if I use our architecture and only take that top pose. And then the green line is our complete method. So it is significantly better than all of the others. And actually, I would argue could be potentially very useful if you think in terms of large scale validation, and also think large scale of virtual screening of trying to find actives within the set. But of course, this is on the cross-validation set. There are other additions we made to the model that I want to just briefly touch on there. The first one is the use of protein family specific models. So there are 102 targets in DUD-E, as I said. 26 of those actually are kinases, and 15 are proteases, 11 are nucleoproteins, and 5 are GPCRs. So the question is, does it help you? And I think it's fairly obvious. It should help me if I have seen a kinase before to predict other binders to other kinases, because they share similar properties. So this is something you would expect to be helpful. Now, if we are working in this machine learning environment, we have sort of 4 choices about how to use that extra data in terms of adding it into the model in some way. The first choice, which is A here, is do nothing, because the kinases are in there anyway. They're part of the data set. So I had all of them, so I didn't have to do anything special. The second choice is just to fine tune what is called the classifier, the very last layer of the network. That's the easiest thing to do. So you just say, well, I'll train everything as is, and then I'll do a little bit of fine tuning on the final classifier to say, I'm trying to classify kinases now. Does that help? The third choice is to fine-tune all the layers. So you train the entire model with all the data, and then you spend a small amount of time fine-tuning it with examples which are specific to your family of interest. And then the final choice would be to train from initialization like this. The problem with that is obviously you lose a lot of information because there's only a small number of kinases compared to the huge size of the original data set. It turns out the best way of doing this is to fine-tune all the layers. So this is to look at the kinases and train the entire model with everything, and then fine-tune with just kinases across all the layers, and then try and test for other kinases. What I'm showing you here is that area under the curve of the precision recall. Yep. And obviously the higher this number, the better. What you can see is if we look at the blue line, this is no fine-tuning. If I test on some kinases, but I have no kinases in the training set, yep, I don't do very well, okay? I've got an area under the curve of about 0.2. As I add kinases in, I get better. So even without doing anything clever, just having more of that data in the data set that the algorithm is learning from is really helping me. What is very impressive is if you look at the fine-tuning all layers, if I just have one kinase and I know that what I'm looking at is a kinase, then what I do is I can add in that kinase and do fine-tuning, and it almost doubles my performance straight away. And as I continue fine-tuning with more and more kinases, it continues to improve my performance. Now, this is, I mean, I think this is a great way of improving your performance and doing good things, but the thing that is slightly scary about this for me is that that line is clearly not plateauing yet. And we could use 16 of the kinases and still have some that we could test on for doing this so that they weren't the same kinases we're using for train and test. Problem, if I need 16 protein targets already in a family with lots of information on them, this is not going to be that helpful. And it looks like the more of those I have, the better, and the better I would get at this. So that's all been cross-validation, and lots of these tools, even before we got as far as deep learning versions of this, when people were using random forest to predict this, there was quite a lot of large number of examples where they were on the data sets they've been trained on, but were less good when you tried to take them to another data set. So we then tested on the ChEMBL set. So this has about 11,000 protein targets, and there's about 2 million compound records overall in ChEMBL. We used a particular subset of 13 targets. So we had to, in here, there's nothing with greater than 80% sequence similarity to any of our test targets. And also there's nothing with any significant alignment of binding sites to any of the test targets. So anything that we had in that overall DUD-E set, these 13 targets don't look like any of those. They do have some relationship to them, as I will show you, and you can see that as we go through. So how do we do here? Well, you see the same pattern. So my red dotted line is once again the random, the blue is the AutoDocVina, and then we're going up towards DenseFS, which is our method there. But the thing I want you to sort of reflect back on is we're clearly having the same problem that lots of other people have already expressed. So we were having an area under the curve of about 0.4 when we were looking at cross-validation within DUD-E. Now we test on ChEMBL a completely separated set, we're about half as good. There's an area under the curve of about 0.2. So though we are still, doing better than others, we're clearly not overcome this problem of how well we might be able to generalize across sets. So where does the kind of future parts of this go? Well, the exciting part of this is the only input is the spatial representation and the atom typing of the dot complex. So hopefully, if you could take this method apart, and I'll talk about that a little bit in the next section of the talk, you could learn quite a lot about what it's actually learning and therefore what is important in terms of binding. And it works pretty well. It's a substantial method of improvement over all the previous virtual screening methods. It outperformed the baseline CNN on 13 of the 14 ChEMBL targets. So we feel, this is happy, it's worth thinking. The improvements came from several different places, which is what I'm trying to show you on the right-hand side here. So if you move from docking software to that baseline CNN, so the original work, you already get a lot of what the improvement is. You then get some improvement just by the change in model architecture we had. The average scoring gives you a bit more, and the protein family models give you a bit more. We also then do an ensemble of models, which gives you a tiny little bit more on the end. I think the protein family models is interesting it appears the more examples we have, it always does better. But will this ever saturate? It must do at some point, and it will be useful to understand just how far you have to go. And as I said, I think we still have this slight worry that it doesn't generalize that well. And that worry then became very well founded. So there were a series of, I would say, very good papers that were published. They came out across sort of 2018 and 2019. And in different ways demonstrated that deep learning methods in particular, so not all machine learning methods, but deep learning methods in particular, are learning the differences between actives and decoys, not the physical interactions between the protein and the ligand. And what they were really getting at there was that you can see, if you look at lots of decoy sets and compare them to the actives, there are chemical differences between them. They've been tried to be minimized in different ways, but it's actually quite hard to do this. And because the deep learning method, you are allowing it to learn whatever it likes to perform its discrimination, it will pick up on any of those subtle differences and can use that to do separation. I'm now going to show you that we really weren't immune to this problem either. So here I did it, this is a very straightforward test. So I've taken the model that we had, so the one I was previously talking about. I'm calling it original FS here because we haven't done any of the family specific training, because that's something we don't want in here because that would confuse issues that we're looking at. Okay, and what I did is I trained it exactly as I described before. So I trained it with the full protein there and the ligand there, and I trained it using DUD-E. Yeah. And now I'm going to test it, and I'm going to test it in two different ways. The first way I'm going to test it is exactly how I tested it before. Okay, I'm going to give it ligands and receptors, and I'm going to try to get the scores out. And that is the green elements that you can see on the slide here for actives on the left and decoys on the right. So you can see, on average, the actives get higher scores than the decoys do. The decoys tend to get rubbish scores, and the actives tend to get really good scores. Okay, so I would be happy. That seems to work. Now I'm going to do a second test. I'm just going to take the protein away. So if you look at the picture at the top there, no protein, no grey. I'm just going to give the program the ligand. Now, I didn't train it with just the ligand, still exactly the same training. That gives me the distribution of purple scores that you can see on the right-hand side. And what you can see is almost nothing has happened to the scores. In other words, the receptor has no idea, that the program has no idea that the receptor is important. It's not none, there are tiny differences in the scores, but on average, it can separate the actives and decoys with only being given the ligand. I've just realized there's a typo on the bottom of that slide that should say distribution of scores unaffected by removal of protein. Okay. And to put this into a kind of perspective, this is for that baseline, so the David Coe's method, and then our original FS. These are precision recall curves again. And the green is if I have used to test it the ligand and receptor, and the purple is if I give it the ligand only. So you can see in both cases, I do worse if I give it the ligand only, but not that much worse. There's still a huge amount of discriminatory power there. And in fact, in the case of original FS, there is more discriminatory power there with ligand only than there is for a standard docking protocol. So I do better just by doing that. And clearly it should not be able to do this, as in the signal should collapse to zero. There should be no useful information because I haven't told it the protein that ligand is meant to be binding to. I've just given it the ligand. So it quite literally doesn't know what I'm asking it, but still it can tell me if it is an active or a decoy. So how do we get around that? Now, the way that a lot of other people have been discussing getting around this, and something that we have also been looking at, is to try and improve the decoy sets. That's the most obvious thing you would like to do, improve the decoy sets. But that turns out to be a very difficult problem because you're fighting against a machine which is basically will learn any kind of slight deviation you have from the two sets being the same. So instead we decided we could try something a bit simpler. What if I gave it the active, but I also gave it as a decoy? And what I mean by that is if you look at this picture that we have here, imagine that the red is the active in its correct pose in the structure, or in its docked pose in the structure, yeah? What I'm going to do then is I'm going to make a random translation, orientation and conformation of that active, and then I'm going to give that in the training set as a new decoy, because it clearly isn't a binding molecule. So now, if I give it the ligand on its own, it will have seen it both as an active and as a decoy. So it can't possibly learn to separate them out in that way. I'm actually going to give, for every active that I use, I'm going to give it 3 new associated decoys, which will be 3 of these random translations with a new orientation and conformation. And you could see it's perfectly possible for this molecule to end up outside the protein. So you might train it quite literally with the ligand on its own in the same way that we were thinking about before, but sometimes you won't. They might be buried inside the protein, or they might sit on a nice surface point. What happens if you do this? So original FS, that's the picture I showed you at the beginning, where the scores for the actives and decoys did not change. So the green and purple together in the sort of A box there are exactly the same for actives pretty much, whether I have ligand and receptor or I give it the ligand only. If I move to TransFS, things look a lot more like I expect. So the scores for my actives and decoys when I have ligand and receptor are in green, and you can see I still see pretty much the picture I did with the original. Nice high scores primarily for actives, nice low scores primarily for decoys. What is reassuring now is that now when I say, okay, if I give you just the ligand, and you should just give me rubbish scores, it pretty much does give me just rubbish scores. So you can see in purple now, for both actives and decoys, the numbers have just dropped to the bottom. But I should put one note of caution on this. It clearly isn't perfect, because you can still see the active scores are still tending to be a bit higher than the decoy scores, even though they are now all very low. So there is still some form of learning going on about this that we haven't managed to get rid of entirely. If we had succeeded in doing what we're doing, there are a few things I would expect to see. And the first one of those is I would hope that we would generalize better. And by that, I mean, if we were learning things that were about biases within the decoy set, then if I remove our ability to do that, when I move to a new set, it's not trying to, if you like, stratify it based on biases that don't exist in that decoy set, because they have a different set of biases. So I should generalize better to unseen targets. So these are the 14 targets again from the ChEMBL set. And TransFS performs well or better than the original FS on 13 out of the 14 targets. Now this is kind of cool, because the other thing I could have done by doing what I was doing was completely destroy performance, which is what I was worried about, which is I could actually have just made the whole thing be unable to learn the interactions at all, because I've given it something so nonsensical. And the nice thing that we observe is, if you look at this table, so I told you that the kind of targets that were in DUD-E, there were quite a lot of kinases in there and proteases and GPCRs. And you can see, you know, the 23 kinases from the original DUD-E set and the 15 proteases. That's the targets that we had that we were training on. But where we see the biggest improvements for TransFS over original FS is at the bottom of this table on the other targets, which are pretty much unrepresented in DUD-E. So there might be one representative of them. So this pushes us towards thinking, though it doesn't prove it, that we are actually learning more about interactions, and we're learning more about how things bind, rather than memorizing types of things that bind to a particular protein family, which potentially is not a helpful thing to do. The other thing is, I would like to be able to see that it is actually learning interactions. And this is something that is actually quite difficult with CNNs because of the way they are generated. It's not a linear sum of all of the things that go into it. So what we've used here is a technique called masking. And in this case, it's atom level masking. So for every single atom, what we did was we ran the model with everything there, and then we ran it with an atom removed. Now remember, that's totally possible because I never gave this model any idea of the chemical connectivity or that atoms needed to be near each other. So it shouldn't care that I remove, if you like, it's like removing red from a couple of voxels in a photograph. So here it would be removing a carbon from a couple of voxels in what's going on. And the colouring here is the difference in scores assigned between that atom being there and not being there. And obviously this won't sum to 100 because it's not a linear combination, but the higher that percentage is, the more it's making a contribution, you would argue, to the CNN score. We've got just 2 targets here, so one in the top row and one on the bottom row. Things in pink or green say that there's sort of contributions to the score, either in a positive or negative sense, coming from this. If we look at the baseline model, so this is the original model from David Coe's, and actually the same is true for original FS, which is sitting next to it, there is no score associated with anything on the protein there. So there's nothing on that white surface of the protein. But you are getting scores coming from the ligand. So parts of the ligand are clearly helping it to decide whether it should say it was a binder or a non-binder. What becomes interesting, and for us a little bit exciting as we move to TransFS, is now we are seeing scores on the ligand and on the protein, but also in complementary sites. So I'm hoping you can see the little yellow dashed lines on here. Those are hydrogen bonds. And what we're seeing is the hydrogen bond atoms on the two sides being picked out as important in terms of the CNN score, whereas the scaffold part of the sort of ligand is not really contributing a great deal to the score. So I'm now going to completely switch gears. So that up till then, I've been talking about this bind, not bind problem, which is a big part of early stage drug discovery. What I'm going to talk about now is one of the other parts of early stage drug discovery, which is how do you build new chemistry that will bind into that site? So in the previous part, I was saying, just give me molecules and I'll just test them all one after another, bang them through, dock them in, throw them through my software. Does it bind? Does it not bind? Now I'm going to try and design better chemistry, given I know something. And in this case, I'm going to set it out as quite sort of straightforward question that apparently comes up quite a lot, which is I have two substructures within a protein. So these might be two things that have been discovered by fragment-based drug discovery. It might be that you have a complete molecule, but you'd like to change the scaffold between the two ends to make it more soluble, to make it work better. I want to design linkers that will go between these two endpoints that will fit and be, so fit in the protein structure and keep the two endpoints in the right positions within the protein structure. Because I could randomly put things in there, but I want something that will work and will fit. There are two main ways people have looked at this kind of molecular generation problem, so generating new molecules in the deep learning space. One is almost arbitrary molecule generation. And there's been a lot of papers where they have demonstrated that you can train, and it's either from SMILE strings or from graph representations of molecules, train it on a large data set like the ZINC database, and then it will pump out things that look like real chemistry and look like molecules. So that's like, these methods can learn to build things that are chemically sensible. Then the other set has been in the area of molecular optimization, where I start with a molecule which has a particular property, for example, synthetic accessibility, and I train a method to optimize that molecule for better synthetic accessibility or any other property that I'm interested in. We want to sort of, I guess, work in more in that molecular optimization set, but what we're interested in is how we do that in a three-dimensional space. So thinking about this as in, these molecules need to bind into a protein, so I need to say, how do I make sure this will fit in the binding pocket of the protein? So this is kind of a picture of what we're aiming to do. On the top is one example of things this could be used for, like scaffold hopping. So here you have two fragments of a molecule, and that red part in the middle is the scaffold. And for reasons to do with maybe it's how it might translate or what exact conformations it prefers, you might wish to change that. And so can you change that central segment, but keep those two other pieces in the correct orientation so they will still bind to the protein? And the second is the kind of structure-based drug discovery, where you have a couple of fragments that you have found bound into a structure, or you found these through other methods, you know where they go, and you would like to be able to build a linking between them in a way that will still fit in the structure and will keep those two fragments in their correct orientations with respect to one another. Now, the standard way of doing this is to have a giant database of linkers, so bits of chemistry, and then you just stuff them in between the two, and then you try and filter it out, you try and dock them in, you try and play with them to try and make them fit. Here, instead of doing that, we're going to use that kind of linker database to train a method to do this, so it will generate fragments. And these fragments, the two advantages should be that everything we generate, and everything will always be an exaggeration, but a lot of what we generate should fulfill the requirements of fitting in the binding site. We should get decent chemistry. We know that these methods can usually do that. And we should be able to generate things that are not in the database, so novel chemistry, so novel molecules. Because however big your database is, it's unlikely to contain all of chemistry, given the estimates of how big that is. So in this, we use graph neural networks. So we're representing the molecule as a graph. So we initialize the two fragments as graphs, as the picture here in the sort of #1 box shows. And then we pass them through a gated graph neural network to kind of encode them. And then we do this initialization step, an expansion set of nodes. Now here, what we have is a set of nodes, and that number of nodes can be set by the user, or you can just run it for lots of different numbers. We've also done tests to show what the optimal numbers are to get certain distances and angles if you'd rather just use that. And then the program itself runs over it again with the gated neural network and takes a, if you like, a random selection so that they are given atom types. And then it builds edges in a stepwise fashion. So it builds an edge from one terminus onto another node. And then the edge is labeled, because that edge could of course be a single bond, a double bond, and the character of that. And then you update, you keep going until you have a completed molecule. Now, what this means is that the number of atoms in between is set as the maximum number I've given as expansion nodes. Sometimes you won't use all of them, as in this case, sometimes you will. To try and put that into what we were actually doing as we did this, we trained with a set of fragments and structural information, so two ends of the molecule. I'll explain exactly where the data set came from, and we gave it the two ends of the molecule and this distance and angle between those two ends. And then we also gave it linked fragments. We represented both of these as graphs, and you should, those little kind of bar charts that are sitting next to them are not quite stacked charts next to them. That is the encoding of a specific atom. Then you encode these, you combine and decode. And in doing that, when you give it the correct answer, you can average and compress that and combine it in. So you're training it to hope that you get the linked fragment back when you do this. And then in the generation step, the input will be the fragments and the structural information. And instead of adding in something which comes from the right answer, in this case, the linked fragments, what you do is you add noise into the system at that point, and it decodes to generate things that should fulfill the requirements that you set in the first place. Training set here, well, unfortunately for us, somebody hasn't solved thousands and thousands and thousands of structures with nice ligands in where we've got good distances and angles. So in order to train this, we took the 250,000 data set from ZINC that's been used by lots of other people. We generated conformers for this. So we just generated conformers. So these are not crystal structures or anything like that. And we snipped up the molecules. So we snipped them so that we could have distances and angles between them. And the cool thing about this is we set some minimum sizes for the linker, and we also set some minimum sizes for the fragments. But of course, we could get multiple SNPs out of a single molecule quite often as well. As you can see in this example here, 2 different SNPs from one molecule. So it gives us a very large amount of training data to train our system on. So does this work? Well, first of all, I want to show you that the structural information is important. And the easiest way to do that is to think of a very simple case. If my linker is basically just a ring, I can have 3 different substitution patterns on that. So I can have pieces hanging off it in 3 different places, in the ortho, the meta, and the para. And those have different distances and angles between the pieces that will be hanging on the end. So there's a variation in distance from just under 3 angstroms to about just over 5.5, and a distance in angle, obviously, from about 60 degrees to about 180 degrees, fairly obviously, as I move between those 2 pieces. So just to check that the system had actually learned anything about this, we used this. So we had done all the training as I've just described with the ZINC set, and then we're going to test just by asking it to, giving it 6 atoms every time, but giving it different structural information. And you can see what happens. The vast number of 6-membered rings generated by Delinker match the substitution pattern that we were hoping we would get just by giving it the distance and angle. So we got sort of 91% ortho in the first instance, and 98% para when we were trying to get para. So we see exactly the pattern we would expect. And also, I think quite nicely, the sort of bleed slightly into the ones that are the nearby cases and no bleed into the ones that are further away. So how did we test properly? Well, we tested on a real set of crystal structures. This is from the CASF set. We've got 250 diverse binders. They're in their active conformations, so these are no longer predicted conformations here. And we're just going to snip out a piece of them and then try and fragment link between them. I'm then going to show you a couple of example case studies after I've shown you the general results to show it can really be used in anger. How do we measure success? Well, the first ways to measure success are in 2 dimensions. So the first thing we want to check is if a molecule is valid. In other words, does it have valid chemistry? So that's the first check. The next check we're going to do is it unique? Because this method isn't very useful if I ask it to generate 500 linkers and it generates 500 of the same thing. Yep, I would rather I was generating lots of different linkers to be able to think about. The next one, is it novel? So is this something that was not present in the database? Because once again, if this was all present in the database, okay, this might be faster because I no longer have to kind of check that it will give me the distance and angles I want by generating conformations, etc. It would still be much better if it was also generating novel chemistry, because then I know that I can go beyond the database of the 250,000 that I have used. And then finally, in this case, does it recover the answer? Since we know what the answer is for the CASF set, did we actually manage to recover the correct answer? And there's a set of 2D property filters we look at as well. So is it synthetically accessible? Is the ring aromaticity all right? And there's also something called the pain score. So how did we do? Well, we did kind of a bit of cross-validation on the ZINC set. We had a small held-out set there to see how we could do, and then you've got the CASF set, and then CASF linkers greater than 5 atoms on the right-hand side. And that's probably where it's most interesting. It's not that hard to work out how to connect things when there are only 3 atoms in between them. There aren't also that many choices. So if I look at the CASF over 5 atoms, if you go from the database, almost everything you pick is valid. I find it interesting that there are some things in the database that are registered as not valid when you put them through testing software. The first thing that is really exciting for us is we tend to generate this very high proportion of unique molecules. So there's lots of different things being generated. And we would expect that because longer than 5 atoms, there are lots of choices that can be made. The next stage, we're generating a lot of novel molecules which pass all of these 2D filters. And then we're recovering a lot more of the right answers than CASF did. Now remember, the right answer is probably just one instance of how you could get between these 2 points, but the fact that we recover it frequently is a really useful metric for us. And do we pass the 2D filters? Well, we do. We do quite a lot, but we don't every single time. But to be honest, the 2D filters, the sort of 2D metrics are not really what we were interested in here. We were much more interested in the 3D metrics, because actually that's what we're trying to do. We're trying to make molecules that will fit into the 3-dimensional shape, and that's what matters here. So we use 2 ways of looking at this. The shape and color similarity score. So that's based on a set of feature maps and molecular volume similarity, and the root mean square deviation, so to check we've got physically the same shape. So if I kind of look at the picture on the left-hand side, so A in the pictures here, we can look at the shape and color score of the whole molecule, or we can look at the shape and color score of just the fragments. So the shape and color score of the whole molecule kind of tells me if I've recovered an absolutely identical molecule. So a score of 0.7 is pretty high, as you can see, but you can see I've got one carbonyl, which I have not got in one molecule, and I've got it in a different spot in the other. What we're really interested in a lot of the time is the shape and color score of the fragments themselves, when here we get a score of 0.92. So all I'm doing there is comparing, are the fragments basically right on top of one another? And it's also what I can compare with the RMSD. So can I produce things where the fragments that I wanted to be in these particular positions are still in those positions? And then you can also check whether that molecule in the middle is similar to the original molecule or whether it is very different. So how do we do? Well, on any measure at any cutoff, we produce a higher proportion of 3D similar molecules than using the database method. And it becomes very noticeable once you start looking once again at these longer, sort of longer than 5 atom things, where the database is starting to really struggle to find anything which is similar, anything which would actually place the fragments in the correct 3-dimensional positions. Whereas Delinker is still not producing that 100% of the time, but we have a high proportion of things which allow you to have these things in the right places. And so obviously you would have far more chance of finding a really good linker by doing this. I'm now going to talk you through a couple of case studies to try and convince you this really does work. So the first of these is a set of molecules bound to a particular crystal structure. So this was a study done by Trapero et al. My brain has now done that completely blanking thing about what it's binding to. So these are, let's come back now, inhibitors of inosine monophosphate dehydrogenase. And actually what they had done is they had actually the fragments, as we show you there, and then they had spent a lot of time with database studies and chemists looking at it and designed a set of linkers. So they actually found 3 linkers that they liked. Only one was, do we have a crystal structure of, which is the one I'm showing you on the right. Delinker recovered both the actual hit that they discovered and the other 2 potent hits that they had in their list. So when we ran it, and we got all of these back. We can also show you what happens in terms of how well we did. So if we look at that shape and color score for fragments, we can see that Delinker is producing a much larger number of fragments, which are much larger number of linkers, which should actually fit very well. We took the kind of top set of those, the database and for Delinker, and did docking with them and looked at the docking score. So the lead had a particular docking score, and we were trying to measure whether we did better or worse than that. To give an idea of whether these hits, you know, we might have more things that look like they would fit, but would they also, do they dock well if we try and do that? Do they have reasonable energetics in the system? And it turns out we're producing a very large number of things that have better Wiener docking scores than the most potent reported binder. A second case study, and this was something we thought would be highly challenging, was to see if we could recapitulate PROTACs. So PROTACs are long, and this is much longer than almost anything we trained on. So we were a little bit nervous that this really wouldn't work. So we've got here PROTACs for a bromodomain linked to a ligase. And we took the 2 separate pieces, and once again, we tried to generate the linkers between them. And the interesting thing here was we decided to generate a much larger number than we normally would. So we generated 5,000 linkers here using Delinker, but that cost in computational time there is very, very small, so it is not an issue to do that. 3,000 of those 5,000 were unique and passed all the TD filters. 2,000 of those unique compounds had the shape and color score better than 0.85. So we got a very large number of things that actually would be able to make that shape really nicely. The images here show just 3 of the linkers that we found, and they all seem able to accurately recapitulate the linker geometry observed in the PROTAC 2. In all the cases, the aromatic system aligns with that of PROTAC 2, and that means they probably will help with having the interactions with that lipophilic interface. I'm hoping that those case studies have helped to convince you that you can use these kinds of techniques, not just to do tasks where I think people felt they were great, which is classification tasks of bind, not bind, though I think there are still things we have to worry about in that system, but also in cases where you want it to build novel chemistry inside a 3-dimensional object that will retain particular 3-dimensional positions and help you build specific chemistry within them. So where do we go from here? Well, I think it's quite exciting that something like Delinker is able to make these unique novel linkers that fulfill the structural criteria. I think what we are working on very hard now is richer input representations. What we've done so far is very basic. We're now looking at how you put the pharmacophores in within this, or even actual 3-dimensional models of the protein. That's quite hard to feed into these types of models. And we want to keep improving how well it does, because I don't want to have to keep filtering out stuff that's not as good. And you can think of other design tasks like macrocyclization. You can think of growing from a single fragment into the protein structure. You can think of how you'd work this with matched molecular pairs and things like that as well. I'm going to finish there. I have to finish by saying lots of thank yous to a lot of people. So I should thank my entire research group. This is us on our group retreat. You might well be able to notice the very elegant photoshopping at the bottom because not everyone was able to attend. And if you're really clever, you'll spot there is a 2-headed person in the back of the picture as well. I should also remember to say thank you to all the people who pay for the research in my group, listed across the bottom there, and in particular for this, Benevolent AI, Excentia, Medical Research Council, and the EPSRC. And I should also mention Fergus Imrie and Jack Scantlebury, who did a lot of the work I just described in collaboration with Nathan, Mihaela, and Anthony. So thank you very much for your time, and I am now very happy to take any questions you might have.